Cell lines are a widely used model system for studying key aquaculture species. However, there are no available cell lines for any marine invertebrate, including key aquaculture species. Primary cell cultures are often used instead, but methods of use are poorly optimized. In this talk, I will describe a new optimized method for establishing primary cell cultures from the Pacific oyster, a key aquaculture species in Scotland and worldwide. Additionally, I will explore how these new methods can be applied to specific aquaculture challenges, such as disease outbreaks, and also show some of the new and unexpected results from this project.
Hi everyone, so I'm Eilidh Costelloe and my talk is on the functional studies of mucosal gill health in Atlantic salmon. So there is a growing interest in gill health of economically important species such as Atlantic salmon. Numerous pathogens and environmentals contributing to inflammation of the gills in farmed aquaculture species. The underlying causes of gill inflammation are poorly understood, and the term complex gill disease and proliferated gill disease are now being put forward to refer to this multifactorial condition. Whole transcriptome profiling revealed a large number of candidate genes consistently altered expression in relation to the histopathological scores for the gill samples of origin. A panel of significant genes were selected for real-time qPCR assay development. And using a predictive modelling approach, the genes of interest were analysed to determine if gene expression can predict histopathology. The outputs of this research will hopefully contribute to improved salmon gill health management.
Effect of macronutrient concentration on fertility of Saccharina latissima. I'm Maryam MacCorquodale and my PhD project is kelp gametophyte bioreactor for metabolite extractions. Large-scale seaweed aquaculture requires reliable source of seed stock that is available all year round. Large-scale cultivation of kelp gametophyte under red light can be challenging. Macronutrient concentration play an important role in maturity and fertility of the kelp gametophyte. Here we investigate effect of nitrate, phosphate and iron concentration on fertility of Saccharina latissima. Manipulation of macronutrient concentration is an effective method to allow vegetative cultivation of Saccharina latissima under white light.
The production of lumpfish for use as cleaner fish has dramatically increased in the past decade. 2020 production is set to exceed 50 billion juveniles, with the majority derived from wild-caught broodstock. The future for this sector is the establishment of a self-sustaining captive-reared broodstock. However, this is currently limited due to poor egg quality. This study performed a comparative analysis of egg composition in relation to batch quality, examining parameters like fatty acids, lipid classes, minerals and pigments from both wild and captive-derived eggs. This study identified significant deficiencies in captive egg composition that are associated with poor egg quality. A total of 48 nutritional components, primarily within free fatty acids, were identified as being significantly associated with egg quality. The goal of this work is to inform the development of effective broodstock diets to improve hatchery performance, reduce the reliance on wild-caught broodstock, and to open up the possibility of selective breeding of the species. Tune in to the aquaculture sector at 11 on Thursday the 8th of October to find out more.
Hi all, thank you very much for listening to my abstract. My name is Joe Penhaul-Smith. I've just finished my PhD at the Scottish Association for Marine Science, and what I'm going to talk about at the MASTS conference is some of the work I've done looking at using the capacity of many microalgae to be cultured mixotrophically and how we can potentially apply this to develop what we're terming designer microalgal feeds through the capacity of our microalgae to partition carbon in different ways, depending on cultures, and developing a tailored biochemical profile from this. So, if anyone has any questions before my talk, please find me on Twitter, ResearchGate, or LinkedIn. I hope to see you all there.
Hello everyone, my name is Alessandro Laudicella. In this talk, I will consider broodstock conditioning, an important mussel hatchery practice aiming to optimize the gonad condition in the broodstock. Evidencing proxies of gonad ripening is important to monitor the gonad maturation process and avoid phenomena like uncontrolled spawning or pre-spawning atresia. Given the centrality of lipids during bivalve reproduction, here I will integrate CMS-based lipidomics with one histological analysis to evidence candidate lipid markers of ovarian maturation. I hope to see you all at MASTS ASM conference, and thank you for listening.